This large black hole is the nucleolus, is the place within the nucleus where ribosomal subunits, 40S and 60S, small and large respectively, they are assembled in the nucleolus. Now, very often the nucleolus is called a suborganel. And it's called for a reason suborganel. It's not really an organ, because unlike the nucleolus is not surrounded by any membrane. It's just a complex of many proteins as well as a certain portion, specific genes, a certain portion of DNA encoding a certain component we will talk about. They are located within this black hole. Right? Okay, so the nucleolus, a suborganel where ribosomes are assembled. Now, how exactly ribosomes are assembled within the nucleolus? So first of all, remember, all of the ribosomal proteins, all of the proteins, these guys, which eventually will compose the small and the large subunits of the ribosomes, they all are synthesized in the cytosol on those three ribosomes. Now, therefore, they must be, and they are imported with the help of importance into the nucleus. In the nucleus, they're looking for a very specific place, all of these ribosomal proteins, the nucleolus, right? Because they know this is a place where these ribosomal proteins will have a chance to meet with the ribosomal RNAs in order to form those subunits, small and large subunits of ribosomes, right? And within this nucleolus, so they're getting into the nucleolus. Within the nucleolus, there are a certain portion of DNA. Each of the chromosomes kind of extend into the nucleus only a certain portion of DNA mode, right? Specifically, a portion of DNA which encodes for this precursor, therefore ribosomal RNAs, right? And these genes are called ribosomal DNA genes. Genes encoding ribosomal RNA, all right? And only RNA, not ribosomal protein, because there are some other ribosomal proteins. Those are used in order to synthesize messenger RNA and so on and so on, out of the nucleus, right? So these ribosomal DNA genes encode a single, very long molecule of precursor ribosomal RNA, all right? So it has been formed in the nucleus. Now, at that point, the proteins, ribosomal proteins, which enter the nucleolus just will bind to this precursor ribosomal RNA, this giant molecule of RNA. That would be your first step. Now you have the precursor ribosomal RNA bound to many different kinds of ribosomal proteins, yellow, green, and so on and so on. This is different kind of ribosomal protein. Now the next step will be the processing. Specifically, there will be a cleavage in a certain places of this precursor ribosomal RNA. One cleave and another, right? That would lead to the formation of this different type of ribosomal RNAs already complexed with the protein sitting, right? Some of these are called the 18S. Some of these particles are called 28S and 5.8S, right? And so the portion of this precursor RNA which has been cleaved into this 18S fragment will form, together with the proteins attached to that, will form this small subunit of the ribosome. And the small subunit will be then exiting the nucleolus and eventually exiting the entire nucleus into the cytosol as now 40S small subunits getting into the cytosol in order to eventually become a part of this ribosome, joining the large subunits which exit separately out of the nucleolus. So there is only one gene, ribosomal RNA gene, which is not, for whatever reason, is in nucleolus. It's located somewhere else in the nucleus, not in the nucleolus. And this other gene is coding some other ribosomal RNA, the so-called 5S ribosomal RNA. And so this ribosomal RNA just getting into the, within the nucleus, into the nucleolus, joining other ribosomal RNAs and proteins, and forming these large subunits of the ribosome, which is exiting, exported out of the nucleus into the cytosol. So the mission is accomplished. The nucleolus is the place where this multi-step assembly of small and large subunits takes place. Now, it's very important to remember, because eventually it will come out before the first midterm, whether or not you need to know all these romantic names like 28.S. What if you mention 27? Would it decrease your grade? 5 point S, that's really super. 5.7, 5.6 won't work. I will not ask you about any specific names, right? Well, in this particular case, in case of the ribosomes, right? Because at the moment, considering that it's 200 course of cell biology, the details are less important. And the details will come out of your head after the final exam. I know for sure. Unlike you have these random memories, right? You know the movie. Okay. Most of you will forget. What I would like you to keep is always understanding, right? You can forget the names of these or specific names of each of these ribosomal RNAs, but you, luckily, for me, will remember, and for you, will remember how actually ribosomes are assembled, right? Okay, good. Now, yeah, go ahead. No. I mean, you know, it's normally located kind of randomly, absolutely randomly. Also, it's for whatever reason cannot be found in the middle of the nucleus, right? And there is a reason for that. There is a reason why it's not in the middle of the nucleus, but any other place, right? And the reason for that, well, at least kind of a superficial explanation for that, because there are a bunch of other particles which we will not even touch in during cell biology course, which also involve in very important function. Some of these particles within the nucleus, they preserve the integrity of DNA, and this is complexes of proteins. Some other particles involved in splicing, so-called splicing, where different portions of RNA will cleave and then join each other, and so on and so on. So basically, it's extremely well-organized space, where many of these particles, and we'll talk only about the nucleolus, perform their very specific function, right? So apparently their positioning within the nucleus reflect a certain type of rules, right? What are these rules? Why is that certain particles belong in certain places? Nucleolus is not one of them, it's not necessarily only close to the nucleolus. Why is they positioned in a certain places? That's a very good question, but we will not know. The nucleolus will disassemble, not only that, the entire nucleus will be disassembled. This entire nuclear envelope will cease to exist, will be separated into small vesicles, and there will be not such an organelle at a certain point of the M phase when nucleus completely disappears in the telophase, and then it will reassemble again. Yeah, go ahead. That's right. Okay, so the first part, the answer to the first part, yes. They are assembled separately in the nucleus. They're getting out of the nucleus into the cytoplasm. And there they kind of in observation mode. They kind of sitting separately from each other. They don't join each other. The condition, the only condition for their joining each other and forming the ribosome is the small subunits must be attached first to messenger RNA. That would change the conformation of the small subunit. It's capable now, and only now, after being attached to messenger RNA, join the large subunit, they form, they click on the ribosome, messenger RNA start translating. That's right. Yeah. They form the so-called small pool of small and large subunits of ribosomes sitting somewhere in the cytoplasm and wait until the messenger RNA arrives. Okay, guys. Any other questions? All right. Okay. Yeah, and you know, just a brief mention, because sometimes students like really demand very extensive explanation for this romantic names. 18S, where is S from? Okay, S just the units of Svedberg, which is a universal unit in order to reflect the rate of sedimentation, another sense of S, sedimentation of these particles, 18S particle, or say for instance when a small subunit is 48 particle, during centrifugation through a certain gradient of density. And the higher the value of S, 40S versus 60S, the higher the value, the faster these particles move towards the bottom of the centrifuge tube during centrifugation. Still have 5 minutes, 6 minutes, right? Okay, guys. So I will fill the time. All right. So let's talk about this gray portion of the nucleus. This what we already know, the DNA. Remember, most of the nucleus is occupied with this gray substance, the DNA. it's not actually only DNA. This is a structure, a formation, which called the chromatin. And the two major group of components which compose the chromatin are not only nuclear DNA, but also many different types of proteins, right? And so they attach to the DNA, all of these proteins, not covalent, through a certain type of ion-ion interaction or ionic interaction, which we will talk about in a moment, right? But now, if you extract the entire chromatin out of the nucleus, and there is a very simple way to do so, right? Then you can measure the weight using a balances, right, of this entire chromatin. And you would find, okay, so the total weight of the chromatins that are extracted from the nucleus is 3 tons, right? So you already know that this chromatin composed of 2 different types of molecules, DNA and proteins. Now we would like to strip out all of the proteins out of DNA, separate these two, DNA from proteins, right? You separate them, again, using a very simple approach, a very simple combination of solvents, and then you measure the weight of the DNA portion of the chromatin and the protein portion of the chromatin, and you will find out that actually the chromatin composed of 2/3, by the way, of proteins and 1/3 of DNA. So the chromatin actually is the very abundant number of proteins, different kind of proteins, and also 1/3 of the chromatin, by the way, is the DNA, right? And one of the group of proteins that bound to DNA are called histones. And so these proteins, different kind of histones, they have small molecular weight, small as compared to most of the other proteins. Most of the proteins within the cell, if you run, say, SDS, page, denaturing electrophoresis, would be concentrated, they stain proteins, will be concentrated in the region about 60, 70 kilodaltons, 60, 70 daltons, right? These guys, the histones, they have molecular weight from 11 to 23 kilodaltons. And therefore, they are smaller than most of the proteins, therefore, they are small proteins. What is very important about these proteins, that they are extremely enriched in 2 kinds of amino acids, lysine and arginine. Now, the pH within the nucleus is 7. At this pH, lysines and arginines carry what kind of charges? Chemistry. Sorry? Positive. Why? Because of the presence of what chemical group? Free amino group, right? Absolutely. And it's protonated at pH 7, right? Chemists and biochemists. Guys, you will confirm. All right. So they carry this positive charge. Now, on the surface of the DNA, all around this double helix, what kind of charges are formed by what kind of chemical group? Negative charges formed by what kind of group? Phosphate group. All right. So it's very convenient for each of these different histones carrying a significant number of these positively charged at pH 7 arginines and lysines to form this electrostatic interaction, non-covalent interaction, which would attach each of this histone protein to the molecule of DNA and would be involved in the formation of this chromatin. Okay? So this electrostatic attachment of histone proteins is responsible for the binding of histones to the DNA. And the last thing, there are 5 different groups of histones, and they have these romantic names: H1, histone number 1, H2A, H2B, H3, and H4. Extremely evolutionary concern between different organs performing exactly the same function. Being bound to DNA and to make sure this DNA is stable and compact. In addition to histone proteins, there are proteins which are called non-histone proteins. Those are not enriched in arginines and lysines. And those non-histone proteins, unlike histones, they are not small. Some of them have molecular weight around 60 kilodaltons. An example will be, for instance, spermine, one of the most abundant non-histone proteins. Many different types. Unlike histones, which have only these 5 types, non-histone proteins are present in about 1,000 different types of proteins. And they all bind to DNA, this non-histone protein similar to histones, and they all do something to DNA. And what they actually do with these non-histone proteins to the DNA, we will talk about during the next class.
buat ni, imahe pasal apa?